All right, so we're back for another deep dive into some cutting-edge research. Today, we're going all in on genomics, focusing on 5 brand new papers that just hit Genomics arXiv.
Fresh off the press genomics, that's what we're all about here.
Exactly. We're talking everything from cancer evolution to the mysteries of sleep, all based on the latest findings. And as always, our goal is to break down these complex studies and deliver the key takeaways in a way that everyone can understand.
No jargon, just the good stuff. Let's jump right in.
Sounds good to me. First up, we've got some fascinating research on prostate cancer, specifically why these tumors can evolve in so many different ways, resulting in a variety of subtypes.
Right, that's a big question when it comes to understanding and treating the disease effectively.
Yeah.
What makes one tumor take one path and another go a completely different direction?
So what did these researchers do to get at that question?
They took a really interesting approach, analyzing genomic data from a huge cohort of 829 prostate cancer patients.
Wow, that's a pretty significant sample size.
It is. And they were looking for any clues, any genetic variations that might explain why some tumors diverge and others stay on a more predictable course.
And did they find anything?
They did. They identified specific genetic alterations that seem to act as switches, if you will. Some seem to actively push a tumor toward a particular subtype.
Like a signal saying, hey, take this path.
Exactly. While others seem to block that branching, keeping the tumor on a more stable trajectory.
Interesting. So it's not just about what promotes the divergence, but also what might prevent it.
Absolutely. And they also found something else pretty remarkable. Some of these alterations seem to trigger each other.
Oh, like a domino effect. One alteration leads to another, and so on, accelerating the divergence into different cancer types.
Precisely. They found a kind of chain reaction happening at the genomic level. And this has huge implications for how we think about monitoring prostate cancer.
Because if we can understand those initial triggers, those first dominoes, we might be able to catch the aggressive subtypes much earlier.
Exactly. This research could pave the way for more targeted surveillance strategies.
Okay, so moving on to something completely different, we've got a paper on using AI for rare disease diagnosis, which is a notoriously tricky area.
Absolutely. It can be incredibly challenging and time-consuming to diagnose a rare disease, and that's where large language models come in. There's a lot of hope that AI can help speed up that process by analyzing clinical notes.
But I imagine those notes can be pretty messy and inconsistent, not exactly ideal for an AI to work with.
You're right, that's a big hurdle. Clinical notes are often unstructured and full of variations, which makes it hard for AI to extract meaningful information.
So how does this paper address that problem?
Well, they introduced 2 new methods, R-driven chain of thought and CAUT-driven retrieval augmented generation.
Those are quite the mouthful.
I know, right? But essentially, both methods try to improve AI's reasoning abilities by combining 2 powerful techniques.
Okay, break it down for me.
Sure. So chain of thought, or CAUT, allows the AI to mimic the kind of step-by-step reasoning that a human expert would use.
So it's not just blindly crunching data, it's actually thinking through the diagnosis.
Exactly. And then there's retrieval augmented generation, or R. This gives the AI access to specialized databases like HPO and MIMIM.
And those are?
Think of them as giant encyclopedias of genetic disorders and their associated symptoms. So the AI can use ROG to pull in relevant information from these databases while it's reasoning through a diagnosis.
So it's like giving the AI both the ability to think like a doctor and a massive medical library to consult.
Exactly. And when they tested these methods on real-world clinical notes, including data from the Children's Hospital of Philadelphia, the results were quite promising.
So this isn't just theoretical, they actually put it to the test.
They did. And some of the recent AI models, like Llama 3.3 and DeepSeek, showed significant improvements in accuracy when paired with these new methods.
How significant are we talking?
Well, in some cases, they achieved over 40% accuracy in pinpointing the correct disease-causing genes within the top 10 possibilities.
That's pretty impressive, considering the complexity of rare diseases.
It is. It suggests that AI is getting closer to being a valuable tool for rare disease diagnosis. And what's even more interesting is that these 2 methods seem to have different strengths.
Oh, how so?
Well, RAG-driven COIT seems to work best with well-structured, high-quality notes.
So the kind of notes that are clear and concise, where the AI can easily extract the key information it needs.
Exactly. While COIT-driven ARG is better suited for handling longer, messier, or more complex notes.
Which, let's be honest, is probably more common in a real-world clinical setting.
Right. So having both methods available could be really beneficial depending on the type of data you're working with.
Now, for our next paper, we're diving into the fascinating world of sleep and our internal clocks. And apparently, ketone metabolism plays a bigger role than we thought.
This study really caught my eye. They used a novel computational approach, basically machine learning, to analyze the relationship between sleep patterns and metabolic changes in fruit flies.
Fruit fly.
Yeah, they're a great model organism for studying sleep. And one of the first things they found was that the flies' sleep periods seemed to function independently of each other.
So even if a fly had a really bad night's sleep overall, that wouldn't necessarily affect all of its sleep episodes equally.
That's what their analysis suggested,
Yeah.
which is a bit surprising. You'd think that if a fly was sleep-deprived, all of its sleep periods would be affected.
It's like saying that even if you toss and turn all night, you might still have a solid nap in the afternoon.
It's a bit like that, yeah. And this independence of sleep episodes allowed them to zero in on how metabolic changes might specifically impact sleep at different times of day.
Okay, so that's where the ketones come in.
Exactly. They focused on manipulating the expression of a specific transporter responsible for moving ketones around.
Ketones being those molecules produced during fat breakdown, right?
Right. And by altering the levels of this transporter, they observed significant changes in sleep stability and disruptions to the fly's normal day-night cycles.
So it seems like ketone transport can actually influence when and how well a fly sleeps.
That's what their data suggests. And they took it a step further by looking at a fruit fly model of Alzheimer's disease.
So they're trying to connect this to a human health condition.
Exactly. And they found that metabolic interventions like supplementing with a ketone body or intermittent fasting could selectively influence the timing of those day-night transitions.
Instead of just affecting the total amount of sleep they got.
Right. It's like saying that instead of just making you sleep more or less overall, these interventions might shift your sleep schedule earlier or later.
So it's a much more nuanced effect than we might have thought.
Exactly. And it highlights the importance of timing when it comes to metabolic interventions and their impact on sleep.
All right, moving on. Our next paper takes us back to cancer, but this time it's about improving our ability to predict patient survival using the fewest possible markers.
Prognostic biomarkers are incredibly valuable for making treatment decisions. But sometimes, models that rely solely on large data sets can be difficult to interpret biologically.
Meaning they might spit out a bunch of markers that statistically predict survival, but we don't really understand why or how those markers are relevant to the disease.
Exactly. And that's where this new research comes in.
Uh-huh.
They propose a Bayesian Cox model that incorporates existing biological knowledge into the marker identification process.
So it's not just a black box, it's actually using what we already know about the biology of cancer.
Precisely. They use something called a graph-structured selection prior, specifically a Markov random field prior, or MRF.
Okay, I'm going to need you to break that down a bit.
Sure. Essentially, the MRF prior allows the model to consider the known relationships between different types of biological data. So instead of looking at genes and proteins in isolation, it's taking into account how they interact with each other within a network.
So it's like saying, hey, model, don't just look at this one gene by itself. Consider its role in the bigger picture of the cell.
Exactly. And by incorporating this prior knowledge, the model can potentially validate what we already know, but it can also uncover new and clinically relevant biomarkers that we might have missed otherwise.
Because those new markers might be hidden within those complex interactions.
Exactly. And when they tested this model through simulations, the results were really encouraging. They found that it leads to a more reliable selection of important variables and provides survival predictions that are just as good, if not better, than methods that don't use any prior biological information.
And what about the accuracy? Can this model hold up in the real world?
They applied it to real breast cancer data from the Cancer Genome Atlas, and the results were very promising. It seemed to perform well even when our understanding of the underlying biological network isn't perfect.
So even with some gaps in our knowledge, incorporating that prior information still improves the model's performance.
That's a big plus for its real-world applicability.
And for our final paper today, we're tackling a pretty fundamental issue in large-scale genomic studies: unmeasured confounders.
Ah, yes, the hidden gremlins that can mess up our results.
Exactly. These are factors that we might not even be aware of, but they can skew our findings and lead to false conclusions.
It's a big problem, especially in genomics, where we're often looking at thousands of genes or other variables simultaneously.
So if we don't account for these hidden confounders, we might end up thinking a gene is important when it's really just correlated with something else entirely.
Exactly. And this paper introduces a unified statistical framework for handling this issue using generalized linear models.
Which is?
It's a type of statistical model that's widely used in genomics and other fields. And their framework relies on some pretty advanced techniques like orthogonal structures and linear projections.
Sounds complicated.
It is, but the basic idea is to disentangle the confounding effects from the true biological signals.
Okay, so it's like trying to separate the wheat from the chaff, the real effects from the noise caused by these hidden factors.
Exactly. Their framework operates in three main stages. First, it tries to isolate the confounding effects. Then, it estimates both the hidden factors and the primary effects of interest.
And finally, it corrects for the bias introduced by those hidden factors to make the hypothesis testing more accurate.
Precisely. And they've shown, theoretically, that their method can effectively control the rate of false positives, which is crucial for ensuring the reliability of our findings.
So it's not just a theoretical solution, it actually works in practice.
It does. Their experiments demonstrated that this method not only controls the false discovery rate, but is also more powerful than other existing methods.
Meaning it's more likely to find real effects while minimizing false positives.
Exactly. And they even applied it to single-cell RNA-seq data, which is a very hot topic in genomics research right now.
So this framework has the potential to be really impactful, especially as we generate more and more genomic data.
Absolutely. It's a valuable tool for ensuring that our findings are as accurate and reliable as possible.
Well, that was quite a whirlwind tour of the latest genomics research. We covered a lot of ground today, from cancer evolution to AI-powered diagnosis to the surprising role of ketones in sleep.
It's amazing to see the pace of discovery in this field, and it really highlights the increasing importance of computational methods in biological research.
Absolutely. We're seeing more and more sophisticated models that can help us make sense of complex data, integrate different types of information, and even account for those pesky hidden factors that can throw off our results.
And ultimately, all of this research is aimed at improving our understanding and treatment of diseases, which is pretty exciting.
It is, and it makes you wonder what other breakthroughs are just around the corner.
I can't wait to find out.
Me neither. Until next time, this is The Deep Dive, signing off.